Welcome to the Chemistry, Biology, and Math Revision Hub. Today we are doing the Pearson Edexcel International A Biology Unit 1 for June 2022. This is the Part 2 video. I will put the link to the Part 1 video below the description box. Let's begin with question 5. Question 5 says, hair straightening or rebonding is a hairstyling technique used to straighten hair using either heat or chemicals. Hair is made from an insoluble fibrous protein called keratin. They want you to describe two structure features of insoluble fibrous protein. So here, there are keywords we get from here. Remember, they want structure features. They want it's insoluble and it's a fibrous protein. So I began by describing what a protein is, then a fibrous protein, and then what makes that specific protein insoluble. So here I said it contains long polypeptide chains. This part here is to describe that this is a protein. It also contains repeating sequences of amino acids, meaning this is all fibrous proteins contain those repeating sequences of amino acids. And then here I said it has a lot of hydrophobic R groups. This part here is to show you why it is insoluble, because these R groups, if they are mostly hydrophobic, then there is a chance that this is going to be insoluble. Lastly, I say the polypeptide chains are held together by hydrogen bonds to form helices, or these are triple helices. All fibrous proteins, they have that triple helix or triple helices held together by hydrogen bonds. So here they only wanted 2 points for 2 marks. If you had written 2 of these, you would get a full mark. Moving on, they say keratin contains a high proportion of the amino acid cysteine. Disulfide bridges form between the R groups of 2 cysteines. The diagram shows the R group of cysteine. This is the cysteine R group. So here they say draw the complex structure of the amino acid cysteine. So if we have, I want to draw for you the main structure of the amino acid, what we need to have is an NH to this side, the carbon, the R group position there, the hydrogen, and the carboxylic side here like that. So this one here is our R groups. Just position this here, and then you have your cysteine amino acids. So that is what I did here. This is known for all amino acids, and then just position that, which is the R group. And here you would get your 3 marks. Let us continue. So here they say the effect of 2 different temperatures on the lengths of the primary structure and secondary structure of a polypeptide was investigated. The table shows the results of this investigation. Here we see the structure of the polypeptide. There is a primary and there is a secondary. For the primary, the length of the structure at 25 degrees is 505, and at 55, it's 505. A 30-degree change did not change the primary structure. When temperature has increased by 30 degrees, the primary structure remained as it was. However, for the secondary structure, heating from 25 to 50 increased the length of the structure. So it means there is a reason as to why these changes occurred. So here they want you to describe the secondary structure of a polypeptide. So here I said, you guys know the secondary structure is either going to have the alpha helices or the beta plated sheets. So here I said, the secondary structure is made up of alpha helices, or it could be beta plated sheets. These are held together by hydrogen bonds between the amino acids. These hydrogen bonds are between amino acids in a different way, like I'm going to show you down here. Be very specific not to say that these are hydrogen bonds are between the R groups. Here you have to be very specific. They are not between the R groups, but they are between the NH of one amino acid and the oxygen on the carbon-oxygen double bond of the next amino acid in the fold. I want to explain that in detail. I got this image online. So if you have this, if this is what you can see, this is the carboxylic end of one amino acid, and let me use this one here. This is the carboxylic end of one part of the amino acid. There is another amino acid that has NH. So this hydrogen bond between the oxygen on this carboxylic and the hydrogen here is what we call a hydrogen bond. And this is making a spiral structure, which is the alpha helix. In the beta plated sheets, we can see again carboxylic to the amine, carboxylic to the amine. It is not between the R groups. You can see the R groups are not interacting at all. So it's very important that you get to know this. I also wrote some extra information. I say this is the coiling or folding for the primary. Coiling or folding of the polypeptide chain to produce a 3D structure. There are 2 types of secondary structures, the alpha helix, which is coiled, and the beta plated sheets, which is folded or pleated. Both are held together by hydrogen bonds. These hydrogen bonds are between the NH group of the NH group of one amino acid and the oxygen of the carbon-oxygen group in the next turn, meaning as it turns around. I hope this makes sense, and I'll go to the next part. Here they say, explain the results of this investigation. I want to take you back to where the investigation was so that we can be able to reason these results, knowing what we are thinking about. The results are here in the table. Since I have showed you this structure here, you can see the secondary structure of the amino acid is folded or coiled. It means heating to a higher temperature is going to break those hydrogen bonds, making the structure straighten out a little bit more. That is why we can see the length increases. While for the primary structure, heating, there are no hydrogen bonds between the molecules or between the different amino acids. So heating to a higher temperature will not affect anything because it is not going to break the peptide bond and there are no hydrogen bonds to be broken. So the structure remains the same length. So let me take you back to my answer. Here I said in the experiment, the length of the primary structure remained the same, while that of the secondary structure became longer as temperature was increased from 25 to 55. I hope you could see that. Then I went on to say the length of the primary structure is unchanged because the heat did not affect the peptide bonds. Remember, in this primary structure, there are no hydrogen bonds, there are only peptide bonds, and these are not going to be broken. I said also the secondary structure has a shorter length than the primary structure because of the folding. I want to take you back to this point here. You can see initially this one has a smaller length in comparison to that because there is greater folding here. So even if the increase in temperature increases the length, but because there is still some folding, the length does not increase as much as that in the primary structure. So let's continue. Then next I said, on heating, the secondary structure increases in length because the heat increases the kinetic energy, which causes more vibrations, and then the hydrogen bonds are going to be broken. So this brings us to the end of question 5. Let's continue to question 6. Here they say the risk of developing cardiovascular disease or CVD is dependent on a number of factors. A, people's perception of these risks are often very different from the actual risks. A study was designed to compare perceived risk and actual risks. A group of people were asked if they thought they were at risk, this is the perceived risk, of CVD. The same group of people were then examined to determine their actual risks of CVD. And here we have the graph to show the results of this study. So on the vertical axis, we have the number of people, and then we have the risk factors here. There is smoking, there is inactivity, overweight, high blood cholesterol levels, and then the high blood pressure. These are all risk factors for cardiovascular disease. And then from the key here, we can see these are the number of people with the perceived risk, and these are the number of people with the actual risk. If we look at the data, we can see that overall, except here, the number of people with the actual risk are actually higher than those that have the perceived risk. So it means there were fewer people who knew that actually they were at risk of having this cardiovascular disease or being prone to cardiovascular diseases, except among the overweight people. In this group, we can see their perceived risk was actually higher than their actual risk because they were more aware. Since they were overweight, they thought there is a higher chance they're going to have cardiovascular diseases. So let me go down here. They say describe 2 conclusions that can be drawn from this study. From the study, I say generally the perceived risk is lower than the actual risk, except among overweight individuals. I think that is what I've already said. You can see here the actual risk is higher, the actual risk is higher, the actual risk is higher, and it's higher here, except here where we have the perceived risk being higher. So overall, the perceived risk is lower than the actual risk, except among these people. And at the next point I said, being overweight is the most common risk factor since more people have the actual risk. You can see the vertical value, more people had that in comparison to the other groups. And lastly, I said, overweight individuals and smokers are more realistic about their perception of their risk factors. You can see here, the difference between the perceived and their actual risk is lower here as well as here. That is because smokers are more aware and the overweight people are more aware of the potential risks that they face, while people who are inactive, people who have high blood cholesterol, and people who have high blood pressure may not be that much aware of the risks that they have. So let's continue. Here they say, explain how this study should have been designed to allow valid conclusions to be made. For this study to have valid conclusions, the number of people used for each category had to be very large, or greater numbers of people. So let me take you back here. It means with smokers, you had to use many smokers. People who are inactive, create a database that has more number of people that have been used for the experiment in order for you to have results that can be reproducible. So we will calculate the mean and therefore have data that is more valid or more dependable. So I said you use large numbers of people. This will allow for more data to be collected for each risk factor, and that can be as if the results have been reproduced. In other situations, let me say if you're carrying out an experiment to study the effect of maybe temperature on a specific, on respiration or on some enzyme activity, it means you're going to use the same enzyme for all the experiments. Now, for human beings, we cannot control everything to ensure that human being A and human being B are exactly the same, because people are different. They have different DNA. They're going to react to different situations differently. However, we have to make sure we use large numbers of people in order for the results to show reproducibility so that they can be more believable. So next I said you have to educate the people about the risk factors and CVD before the interview in order for them to make informed decisions. If you had interviewed somebody who doesn't know what cardiovascular disease is, what causes cardiovascular disease, they wouldn't know. They would just be guessing because you're asking them to make decisions that are not informed. However, the better way would be teach these people or educate them about cardiovascular diseases. You tell them salt intake, you tell them smoking and inactivity as well as the other things. Then they will be more informed so that when they are making decisions, they will be making informed decisions that are not going to bias your results. Then next I said the people interviewed should have no known underlying health conditions because their perception will not be genuine. If somebody knows already that they have high blood cholesterol, then or if they knew that they have cardiovascular disease, of course, when you ask them, they know they already have the condition. They will tell you the result, and you would think, oh, their perceived risk as well as the actual risk is exactly the same, but they already knew the condition was there before the interview. So you have to ensure that they do not know their underlying conditions. Next here, they say, explain why it is important that a person's perception of the risk is close to the actual value. This is going to be important because if it's close, then it shows that the person is aware of the risk, and then they can be able to make lifestyle changes in order to lower the risk or minimize the progression of the disease. So I said, if they are close, meaning the actual and perceived risk, then it means that the person is aware of the condition or the situation they are in, and they can change their lifestyle in order to lower the risk. So as they change their lifestyle, we can be able to carry out health checks in order to monitor the progress. This progress is due to the lifestyle changes that they have carried out. We will monitor the progress of the results as these changes are being carried out in order to see if the progression is going down or if it's still increasing. And then if it is, sometimes we can give medication. It can be prescribed to slow down the progression of cardiovascular disease. And lastly, I said, if the perception is higher than the risk, let's assume if somebody thinks they have a higher risk of getting cardiovascular disease than their actual risk is, then they are going to make life changes that will be unnecessary. An example, for some people, they will start dieting, they become skinny, and that is an unnecessary change because they do not have a risk, or the risk factors are actually lower than the actual factors. So the actual factors are lower than their perceived factors, or risks, and therefore they're making unnecessary changes. Here they say the 10-year risk of a person developing CVD can be estimated in a number of ways. The Framingham Risk Score uses a point scoring system to estimate the 10-year risk of developing CVD. They say table 1 shows the point scores for different risk factors in women. So let's see. There is an age. This is all among women, those who are aged between 35 to 39, 40 to 44, 45 to 49, and so on. We can see their specific points for the risk factors. The total cholesterol is also given in the various ranges, and we can see their risks, the point scores that they are awarded. For smoking, we can see as those for the specific age groups. And then the systolic blood pressure, we can see the specific blood pressures for the age groups as well as their points that are awarded. So I will come back to these points that I marked on this table for the next question. So let's first continue down to table 2. So table 2 shows the 10-year risk for the total points scored calculated using information in table 1. So we'll calculate information from table 1 and we will refer to table 2. The parts I marked here, I'm going to come back to them when I read the next question. So let's just continue here. They say, calculate the 10-year risk of CVD for a 35-year-old woman, so age is going to be 35, who has total cholesterol. Cholesterol is in that range, and this person is a smoker. So we will look at smokers among this range, and then has systolic blood pressure of 132 mmHg. So I will go back to the previous page. So the information here we can see is this woman is 35 years, so she scores -3 on age. Those are the points. And then for total cholesterol, she has 242. It means it is in this range. So since she's 35 years, she scores 11, plus 11. So she's a smoker who is 35 years, so she's going to score a plus 9. And then for her systolic blood pressure, it is in this range, which is 132. So she scores this because that is her age. Now, when we add up these, -3, plus 11, plus 9, plus 2, we get a 19. And that means the total points scored are 19, and therefore the 10-year risk of developing CVD is 8%. We can see this is going to be the answer for the question on the next page. I hope you have seen, have calculated that. Let me continue. The answer here is 8%. So going down here, they say, explain the lifestyle changes this woman could make to reduce her 10-year risk of CVD. They want you to use information in the table to support your answer. So you saw the conditions this person has. The person is a smoker, and then she has a lot of total cholesterol. The total cholesterol in her body is a bit, not so high, but it's a bit high. And then we have the blood pressure is high as well. So I said she should stop smoking to reduce the damage to the endothelial lining of her blood vessels. Remember, if they are damaged, then atheromas can form, and those can lead to problems or cardiovascular diseases. And then next I said she should reduce consumption of saturated fats in her diet to decrease the cholesterol in her blood. Remember, she has a total cholesterol of 242 mg per decimeter cubed. Also, I said this cholesterol could accumulate and enhance plaque formation, since from your AS, plaques are formed if there is cholesterol and there is damage to blood vessels. Also, increase exercise. She could do that, or she could consume less salt. Take antioxidants. Taking antioxidants, because she's a smoker, smoking can lead to formation of free radicals, or due to particles from smoke, free radicals could be formed, and these antioxidants can be able to decrease those free radicals. So she could take antioxidants, she could increase exercise, and she could consume less salt. So all these points could be acceptable. Next, they say which pair of drugs may reduce the 10-year risk of CVD for this woman. Since this lady has, we can see she has high blood pressure, and then she has, you can say a combination, of course, of medications that are going to reduce this. I just chose the because it is an antihypertensive, and then we have the statins. Statins work by lowering the low-density lipoproteins or low-density lipoprotein cholesterol in the blood. And if this is lowered, it means there is going to be a lower chance of formation of atheromas, and therefore blocking of the blood vessels as well as CVD can be decreased. For this high blood pressure, of course, we know high blood pressure can also damage the blood vessels, leading to atherosclerosis as well. Moving on. Here they say suggest why this method of determining the 10-year risk of CVD may not be accurate. It may not be accurate because these are not the only risk factors. There are other risk factors that have not been included. So I say other risk factors have not been included. Factors like diabetes. If somebody has diabetes, they could have a higher chance. There is a genetic predisposition. Because this person was a woman, being female is not the only thing that is going to cause maybe a change in age with sex, so that maybe you have some CVD as well, or not. But here we have genetic predisposition. If they have genes that can lead to that, we need to know that in the table or in the information. If the person has levels of activity, if the person is active, how active are they? Are they active by just walking, or they go to the gym, or they run? All that information has to be included. And then for smokers, we need to know the number of cigarettes that they smoke per specific time. It could be per day, it could be per week. and how long have they been smoking. We know if somebody has been smoking for a longer period of time, then there is a higher chance that their risk is increased than somebody who smoked beginning maybe last year or yesterday. And also, those who take more cigarettes, they have higher chances of affecting the blood vessels and leading to more atheromas than those who do not. So all this information has to be included. And lastly, here I said, the levels of cholesterol may be estimated and they may not be accurate. I'm talking about those in the table. They might be estimated values, not 100% accurate, and that could lead to this data being inaccurate. So this is the end of question 6. Let us continue to question 7. Question 7, the diagram of a cell membrane determines its properties. The membrane is a phospholipid bilayer. The table shows one way that the components of the phospholipids can be drawn. So they've given us the phosphate, which is circular, the glycerol, which is here, the fatty acid, which is that, and then the bond. So they asked us to draw a diagram of a phospholipid using the information in the table. A phospholipid contains two fatty acids that are bonded to a glycerol that is bonded to a phosphate. Now, the two bonds are here, and then there is one bond here. So if you had drawn it like this, that you get 2 marks for that. So ensure that you put only 2 fatty acids, not 3, like some of you accidentally do. So let's continue. Here they say one of the bonds present in phospholipids is an ester bond, which diagram shows an ester bond. An ester bond is between a carboxylic acid and an alcohol. I'm talking in terms of biology. For chemistry students, you know, there are other bonds that can have the same ester bond. Some other 2 groups, maybe an acyl chloride as well as an alcohol could also form an ester bond. But in this case, let's go to biology, which only talks about a carboxylic acid as well as an alcohol. This one here is not an ester bond because it's between an amine and maybe a carboxylic group. This one could be between 2 groups that may not be very specific. And we can see this one here is not as well. So the only answer is this part could be a carboxylic acid and this part here could be an alcohol, and that is an ester bond. Moving on, they say the fluidity of a membrane is increased by a number of factors. One is increase in temperature. The other is increase in the proportion of unsaturated fatty acids. There is a decrease in cholesterol and fatty acids with shorter chains. All these factors here are increasing the fluidity of the membrane. So a higher temperature increases fluidity. The increase in proportion of unsaturated fatty acids will increase fluidity. A decrease in cholesterol will increase fluidity, and fatty acids that are shorter chains or shorter side chains will also increase fluidity. So here they say explain why an increase in temperature increases the fluidity of the membrane. Remember, this is temperature. Always when they ask about temperature, remember kinetic energy. An increase in temperature increases the kinetic energy, and therefore the particles will be moving faster. So this will cause the forces of attraction holding, in this case, the fatty acids and the other components together to be loosened or to be kind of broken. I say this causes the forces of attraction holding the fatty acids and other components of the cell membrane together to be broken, and therefore the phospholipid becomes more free. Give me a minute here. Or weakened. They can be broken or they can be weakened. And therefore, the phospholipids become more free to move around in the membrane, and this causes fluidity. I think you can see that. So let us continue. Here they say, which row in the table describes an unsaturated fatty acid compared with a saturated fatty acid with the same number of carbon atoms. Remember, a saturated fatty acid has no carbon-carbon double bond, and unsaturated fatty acid has carbon-carbon double bonds. So we are comparing unsaturated fatty acids to saturated fatty acids. What we need to know is, here they ask, has double bonds between 2 carbons? Yes, remember you are comparing unsaturated to saturated. Unsaturated has carbon-carbon double bonds, and the number of hydrogens in unsaturated, we have fewer hydrogens, so the answer here should be a C, as we can see there. Moving down here, they say, explain why a decrease in cholesterol increases the fluidity of the membrane. This is a decrease in cholesterol. Remember, cholesterol holds the fatty acids intact, or it's going to be attached or holding the fatty acids in position. So if we have more cholesterol, it means more fatty acids are going to be held or restricted in motion, and therefore the membrane is not going to be as fluid. However, having lower cholesterol concentration will have fewer molecules that are holding these fatty acids in position, and therefore the membrane is free to a little bit be more fluidy due to the motion of these fatty acids. So here I said cholesterol interacts with the fatty acids of the phospholipids, restricting their movement. A decrease in cholesterol will cause the movement of the phospholipids not to be so limited. Therefore, they can move more, and we can say the membrane is fluid, or the fluidity of the membrane. Down here we say suggest why a fatty acid with a shorter side chain will increase the fluidity of the membrane. I want to take you back here. Remember they said fatty acids with shorter side chains. Why is it that that is going to cause more fluidity of the membrane? One thing we need to know is that if these shorter side chains are going to have four areas of contact. The areas of contact between the side chains and other molecules are going to be fewer if it's shorter, and therefore there will be less intermolecular attractions or forces of attraction between them. And those forces being fewer, it means they're easily broken. Therefore, the whole membrane is going to move due to the shorter side chain. So I say the shorter side chains of fatty acids will form fewer intermolecular interactions. This is very important. And this will require less kinetic energy, so movement will be observable even at lower temperature. The key thing is, if a side chain is shorter, there are fewer areas where it can form forces of attraction or intermolecular forces of attraction. But if they are longer, it means they can form these forces of attraction between other molecules, and the total forces of attraction formed will be more, meaning they will require more energy in comparison to where fewer forces of attraction were formed. So this is going to be more fluidity in comparison to this one that will be only fluid at higher temperatures. So that was what my answer was focusing on. Next, they say, studies have shown that when fish are moved into water at a low temperature, the fatty acid content of their membrane changes. So they suggest these fatty acid content. So they suggest how the content of the fatty acids in these membranes may have changed. This is moving to lower temperature, to water at lower temperature. Now remember, the fluidity, the membrane tries to ensure that it remains fluid, or it increases its fluidity when the conditions change. If you move the fish to lower temperatures, the membrane is going to fight to ensure that it contains or it has the same fluidity in order for molecules to move across it. To do that, it means it's going to change itself to have the factors that increase fluidity of the membrane. One thing that we're going to see is the longer side chains will shorten. So the membrane of the fish is going to have shorter side chains. Remember, we have seen that shorter side chains will have fewer points of contact for formation of intermolecular forces of attraction, and that means they are going to be loose, and therefore they will, at lower temperature, they will not be as strongly bonded to other molecules, and therefore the membrane will be movable, or it will be fluid. The next I said, saturated fatty acids will be converted into unsaturated fatty acids. Again, you remember we have seen the factors that maybe help the membrane to be more fluid. One was having unsaturated fatty acids, so this is going to be what we'll observe. Down here, I tried to include the factors from the real paper. They said increase in temperature will make the membrane more fluid, increase in the proportion of unsaturated fatty acid. This is going to increase fluidity, as well as the fatty acids with shorter chains. So I marked these 2 points because they correspond to the question we are trying to answer. And then here I say the membrane will still possess its properties to carry out its function without interruption. This is if the temperature is changed. Remember the conditions here we are looking at here is when the fish is moved to lower temperatures. The membrane should be able to perform its function in order not to starve the cells of this fish, so that everything can be okay. Lastly, they suggest why these changes are necessary for the survival of the fish. I said the side chains become shorter so that the fluidity of the membrane is maintained. If they do not shorten, fluidity will decrease and the molecules will not be entering or leaving the cell appropriately. This ensures that the membrane retains its shape so that the processes like exocytosis, endocytosis can still occur around the membrane, so that important substances are moved across the membrane because the fish needs to survive. So this brings us to the end of question 7. I'll move on to question 8. Question 8, Mutations can give rise to cancer or genetic disorders. Cancer is one of the main causes of death in the world. In 2018, in the UK, there were 541,589 deaths in total, and 166,800 of these were due to cancer. They want you to calculate the ratio of deaths caused by cancer to deaths not caused by cancer. And they want you to give your answer in 2 decimal places. Remember, this is death caused by cancer to those not. So the death caused by cancer is this because they say that of those were cancer. So that is caused by cancer, meaning the death not caused by cancer should be that minus that, which gives us 374,789. And then now to find the ratio, it's going to be that to that, which gives us 0.45 to 1. And therefore, I wrote my answer like this. Although if you had also written 1 to 2.25, that could also be acceptable. So here they say scientists have identified many mutations in cancer cells and are trying to identify the mutations that are significant. The chart shows the proportion of cancers caused by some types of mutations. So when we see the pie chart here, we can see type 3 mutations, we can see the biggest percentage, then type 1 substitution mutations. These are also substitution mutations. We have type 2 substitution mutations and the other types of mutations. So let's go down here. They say name 2 other types of mutations. Remember, this is substitution, and substitution. It means the other 2 I could write are insertion mutation or a deletion mutation. So this could be okay. Moving on. Here they say estimate the percentage of cancers caused by type 3 substitution mutations. So here, this is an estimate. I know most of you are not going to have a protractor in the biology exam. So when they say estimate, it means you're going to try to guess the percentage. So let me take you back. I guess the percentage by, you have to know that this, when you break it down like that, that is about 180 degrees. And now this is going to be about 180 plus about 45 or something like that. So here I said 180 plus 45. It gave me my estimate of the percentage to be 225 degrees. And since this is 225 out of 360, the whole pie chart is 60 degrees. So 225 out of 360 times 100 will give me the percentage of that specific part, which is a type 3 mutations. I want to take you back. I'm looking at these mutations here. If I estimate this to be 45 and that is 100, that total is going to be 225. Now moving back. So the percentage I got is 63%, and that was my answer as we can see here. Now I wrote something for you. This angle is an estimate. However, if you had a protractor, you could be able to get the exact angle. They call it an estimate because they know most of you are not going to have protractors. Next part says, by the way, the actual answer was between 58 to 67%. All these answers were acceptable around that range because they know students are not going to guess it correctly. There is some ranges that are going to be erroneous. So they say a type 1 substitution mutation in a gene alters the DNA and messenger RNA, but does not affect the protein synthesized from the mutated gene. They want you to explain how a substitution mutation can alter the DNA and messenger RNA, but not a protein. We expect most substitution mutations to actually alter proteins, but if it doesn't, then it means there is something that worked. So a substitution mutation replaces one base. So if a base is replaced in the sequence, remember those bases are going to be maybe A, U, G. Let me give another example. Let's say UAA, UAC, UAG, or something like that. If this is what we have in the messenger RNA, and this is what is going to have, I'm using uracil, so let me go back to the DNA. If this is UAA and UAG in RNA and UAC, it means here we're going to have, of course, thymine. Here we have, thymine, and then we can have a combination. So it means, and if this one here is changed, maybe to C, we are going to have a G here. But one thing we know about DNA or about the genetic code, we know that it's redundant or it's degenerate. It means most of the time it's just the first 2 nucleotides that are going to be coding for a specific amino acid, not all the 3. The triple codon does not all code necessarily. It's the first 2 within the codon that are going to be significant. So here I say, I want you to listen to me carefully. A substitution mutation replaces one base in the sequence of DNA. If one base is replaced, it means the other two are going to be correct. So when they are transcribed to messenger RNA, the different base is inserted in that specific location. However, we need to know that the genetic code is degenerate or redundant. That means only, I say it here, it means because a single amino acid can be coded for by more than one codon. So because the first two letters are the ones that are significant, so more than one codon can code for the same amino acid. So the resulting codon will code for the same amino acid. If it does, and it codes for the same amino acid in the non-mutated, as it would be in the non-mutated DNA, then it means there is no change that has occurred, and therefore the produced protein is going to have the same amino acid in the same location, because the change that occurred due to the mutation changed the base, but the resultant maybe codon in the messenger RNA also coded for the same amino acid. Therefore, the protein will not be changed, its structure will not be changed, and its function will still remain the same. I hope you get that. So moving on. Here they say, phenylketonuria or PKU is a genetic disorder. This disorder is inherited in a similar way to cystic fibrosis. The pedigree diagram shows the phenotypes of individuals in one family affected by PKU. So here we can see the phenotypes. These are not affected, these affected. The key thing here is if two unaffected parents produce affected children, then we know there is a chance that these are heterozygotes. So I can see, looking at these two, I know these are the heterozygous parents for the condition. And they ask, what is the probability of couple 1 and 2 having a fourth child that is a male affected by PKU? Now here we have to know probability of male first, and then you multiply that with probability of a child having PKU. Now these parents here, I looked at the probability for the sex of the child. A mother, a father, there is a chance that they can have 2 girls or 2 boys. So it means having a male is going to be a half. That is the probability of male. Then for the genetic condition, I let it to be H. If they are both heterozygous, like we see here, there is a chance that is a 1 in 4 chance that they're going to have a child who is affected. So that probability is 1/4. Now, since they asked us to find the probability that a male child is affected, it means probability of being male times the probability of being affected, which is a half times a quarter, giving us 0.125, and the answer ends up being B for that. Let us continue. So here they say, discuss the extent to which this pedigree diagram can be used to identify the genotypes of all the members of this family. I want to take you back to this pedigree diagram. Remember, individual 1 and individual 2 have individual 3 and individual 4 as children. Now, although individual 1 and 2 do not show the characteristics or do not show the phenotype of this condition, we see that individual 3 and 4, who are children of individual 1 and 2, have this condition. This shows that their parents are heterozygous for the condition. So now we can know the genotype of this, and that is heterozygous. Now, when we come here and look at individual 5 and individual 6, we cannot tell because we do not know the family history of individual 6. And we are sure that the individual 5's history has that specific gene or allele for having this genetic condition. However, because they are not expressing this condition outwards, they may be homozygous dominant or they may be heterozygous. We cannot conclusively tell. Now, when we look at the children from individual 5 and 6, we can see that they are all not showing the phenotype for that specific condition. And it could be that individual 6 is homozygous dominant if individual 5 is heterozygous, so that all the children are always going to show no condition outside, even if some of them might be heterozygous or they could be homozygous dominant. So we cannot tell. For this one here, we only guess, yes, maybe they don't have it, but because their family history has that condition, there is a chance that these children are heterozygous, or there is a chance, but actually these children are homozygous, so we cannot conclusively tell the full genetic detail of individual 5, individual 6, individual 7, individual 8, and individual 9, but we know 1 and 2, and we know 3 and 4. So let us continue back. Here I said, Individual 1 and 2 must be heterozygous for the condition because they did not have PKU, yet they had children with PKU. These children are individual 3 and individual 4. And then moving on, I say individual 3 and 4 must be homozygous because they have PKU. This is homozygous for the condition. Okay. Then they have PKU. They must have received both alleles from one of their parents, since both alleles must be recessive for the condition to be expressed. So they are homozygous recessive. Now, for individual 5 and 6, we cannot tell if they are heterozygous or homozygous dominant because these two genotypes will give rise to an individual without PKU phenotypically. Also, even though we do not know any information about individual 6's family, lack of PKU among her children, who are individuals 7, 8, and 9, makes it less likely that 5 and 6 were carriers. Carriers meaning they had that specific allele. Lastly, I said for individuals 7, 8, and 9, we can't tell if they are homozygous dominant or heterozygous. However, because there are alleles for PKU in individual 5's family, individual 7, 8, and 9 could be carriers. We cannot conclusively tell. So this brings us to the end of this paper. Thank you for being with us. Please do not forget to subscribe to our channel. See you in the next video. Bye-bye.